Hello everyone. In this lab, we will learn how to conduct a biochemical oxygen demand test using a dissolved oxygen or DO meter. The biochemical oxygen demand, or BOD, is a measure of the amount of oxygen consumed during the biological degradation of organic material in a wastewater sample. Because oxygen consumption is directly related to the amount of organic material in a sample, BOD is widely used as a measure of the organic strength of wastewater treatment plant effluents and to evaluate the removal efficiency of organic compounds in biological waste treatment processes. Since the completion of this process would take a considerable length of time, it is mostly common to measure the DO consumed over a 5-day period, also known as BOD5. Due to the lab schedule, we will use BOD7 instead of BOD5. Instead of cultivating the sample for 5 days, we cultivate it for 7 days, so you can come back next week at the same time. Before we start the lab, I would like to remind you of some safety guidelines and to please wear all required PPE. PPE includes safety goggles, a lab coat, gloves, and tight long hair. Glass bottles may cut fingers, so always hold them in the middle. Be careful of any water on the floor. Walk slowly in the lab. Treat simulated wastewater and seed inoculum as though they may contain biohazardous material. We can now move on to the actual procedure of the lab. To begin, we will use the simulated wastewater, SWW, as our sample in the lab. Label 36 BOD bottles with group ID, sample ID, and analysis day, either DI or DF. Please refer to Table 1 for this. Transfer GGA or SWW to the appropriate BOD bottles with the designated volume. Transfer 2 milliliters of seed to each BOD bottle, excluding the dilution water DW check. Carefully fill each BOD bottle to the middle of the neck with dilution water, and be careful not to allow any turbulence or bubbles to form in the bottle. Gently insert a glass stopper and mix by slowly inverting 3 times. For the DF labeled bottle, we will put a plastic cap to water seal it in order to prevent the oxygen transfer between the inside and outside of the bottle. Then, transfer them into an incubator to cultivate them at 20 degrees Celsius for 7 days. After the preparation of BOD bottles, we can determine the initial DO, DI, using the DO meter and record data. Note that all DO meters are calibrated by the technician. Put the bottle in the holder, which is attached with a DO meter. Rinse the bottom membrane part of the DO meter with deionized water. Open the sample bottle stopper and put in the DO sensor quickly and gently. Make sure to not introduce any bubbles. Turn on the stir by switching the red button. Wait about 5 seconds and read the number. The DO reading may not be stable, so just take the reading at 5 seconds. Stop the stir, take out the DO sensor, and rinse it. Put it back in the DO sensor storage bottle. Next week, when you come, take the DF bottles out from the incubator and repeat the previous procedure to determine the DF. Lastly, calculate the 7-day biochemical oxygen demand of the simulated wastewater, the blank, and the standard using the equation shown here. After this lab, you should know how to measure the DO using the DO meter and how to determine the BOD of a water sample.